Good afternoon, everyone. I'm hoping I can make it through this talk. I'm allergic to guinea pigs, and we have 2 guinea pigs at home. And every once in a while, it just like flares up. And right before I left home, I was like, oh God, it's flaring up. Does anyone want guinea pigs? Been trying to get rid of them for a while. Anyway, I got my water, hoping I could get through this. So today I'm going to talk about soil-borne pathogens and cover crops. And since it's Valentine's Day today, I thought I would start out with professing my love for legumes. Yeah. Like, don't tell my husband. I don't even think I've said Happy Valentine's Day to him yet. Yeah, I love pulses, I love legumes. I think we all know why they're beneficial to our agricultural industry, particularly because of that picture that you can see there on the left, those beautiful nodules that they produce. And I primarily work as a pulse crop pathologist. Usually I'm up here talking about root rots, and you haven't escaped. I'm still going to talk about root rot of pulses, but I'm also going to talk about legumes. I just wanted to start out, with this idea that all pulses are legumes, but not all legumes are pulses. So when we talk about pulses, it's really the legumes that produce the dry edible seed that we eat. And then we have a lot of other legumes that can be part of a cropping system, particularly as they're used in cover crops. So why do I love pulses? I love pulses because they're really good for the environment. They have ecological pest management services, which I'm going to talk about on the next slide. They have sort of this smothering effect on weeds because they tend to form these dense canopies. They provide improvement in soil properties, improved soil microbial ecology. Really, it's that nitrogen fixation, those beautiful nodules that I showed, enhance resource efficiency, and reduce energy use. And they're an essential component of crop rotations and sustainable agriculture. And I think we all know, we're all in this room because we're interested in farming smarter and ways that we can improve our cropping system. And we all know that crop rotations are essential to sustainable agriculture. They reduce the incidence of insects, plant pathogens, weeds, nematodes, and of course, the greater the difference between crops, then the greater the effect that you get from having these crop rotations. And so what we tend to have is an oilseed, cereal, and pulse crop rotation in our main cropping systems. But pulses are still a very, very small percentage of that. So it means that we're not seeing that kind of 1 in 4 pulses that the pulse associations, the pulse growers industry would really like to see, where we see 25% of our agricultural land cropped to pulses. And that's really when we'd start getting that benefit of that nitrogen fixation of pulses. But there's some challenges with pulses, and I'm going to kind of talk about that in the next couple slides. So first, let's kind of take a step back and look at what is a healthy soil. I'll admit I wasn't able to be here for most of the day, primarily because of this kind of allergy attack this morning. So I don't know how much this was covered already, but when we look at sort of what the definition is of a healthy soil, it's the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So very, very broad definition. Basically, it's saying, can your plants grow in the soil? Then it's healthy. But we have all these different ways that we measure healthy soil, including soil organic matter, all the different physical properties like the bulk density, compaction, porosity, a lot of the chemical factors. We're looking at EC, we're looking at pH. And then there's the huge biological component. And that's where there's really a lot of research interest right now, is trying to define what is the biological component of a healthy soil. And then the literature really tends to agree that cover crops are a major factor, a major driver in producing these healthy soils. This was a review that was just published in 2023 that really looked at a kind of a meta-analysis of what are all the ways that cover crops can improve your soil. And if you'll notice in this picture, this is actually just the graphical abstract from their research paper. You'll see that they're right away showing 3 different legume crops to be used as cover crops, because we want that benefit of legumes, so we're finding that they're used more and more in cover crops. And then what we can see kind of on the far right, they're trying to sum up everything that they found in this big meta-analysis of cover crop papers, is that we can see that you get increased saturated soils, you get better infiltration rate of the water in your soil, sort of better porosity, reduced compaction, better nitrogen use, and that really comes from having legumes as a cover crop in these systems. Increased soil organic carbon, again, and a lot of this is because of legumes as cover crops. So I was also interested in what cover crops are being grown in the prairies. If I'm here to talk about, what's the potential for crossover of pathogens from your cash crop to your cover crop, then we need to know, well, what are people growing as cover crops? So I got in touch with Yvonne Lawley from University of Manitoba, who I think Farming Smarter has done some work with her, and asked her about she had done a prairie crop survey in 2020, and she was kind enough to share those results with me. And so this is just a map of showing where farms, in their survey in 2020, I think they surveyed 280, they had 280 respondents. This is showing a map of all places that grew cover crops. And we can actually see that down in southern Alberta, those darker blue squares there was a lot of farmers that did grow cover crops. A little more scattered throughout Saskatchewan, and then in Manitoba as well, a fair number of farmers are using cover crops. And so what cover crop species are primarily being grown? And this was very interesting to me because we find a lot of legumes on here, and it's what you would expect when we know how legumes are contributing to soil health, and that's why we want to use cover crops. And so number one is really any kind of clover, and then if you go kind of fifth on the list there, we can see peas, immediate red flag for me, and I'll talk about why, like I said. Kind of a spoiler, I like root rots, and then you say, oh, you're putting peas as a cover crop? Like, uh-oh. And then if we go down the list, we can see other legumes there, alfalfa, sweet clover, some vetches, faba beans, lentils are kind of at the bottom of the list. And then we have kind of the second most is fall rye. So I guess a cereal, but it's not really grown for grain. That's a really, really common cover crop, and I'll cover that closer to the end of my talk. And then we see kind of oats are pretty common as well. So just kind of an idea of, okay, what are producers putting out there as cover crop species? And if you want more information, Yvonne gave me this to put up. I think if you can scan the QR code from there, then you can actually dig right into their survey report. And there's a lot more information about cover crops there. And then what was interesting is that from that meta-analysis review that I showed you that had the nice graphical abstract, they also sort of broke down what the different cover crop species were being covered in all these research papers. Don't ask me about the organization of their graph on the top there. If I was writing that paper, I would not organize it that way, but we could kind of see that there's grasses are green, and so rye and just grass are kind of the two highest bars. And then legumes are blue, and then we can see that vetches are actually quite high, kind of globally being used as cover crops. And so if we look at the bottom, that's kind of summarizing all those different crops. We can see that sort of number one was in the group of sort of grasses, and then number two is legumes, and then they categorized everything else as non-legumes. But anyway, so that's sort of matching what we were seeing in the prairies in terms of what's being used as a cover crop. So if we're kind of putting all this together of, okay, cover crops are good for soil health, pulses and legumes are good for soil health, then the question that I like to ask my soil health scientist colleagues are, can soil-borne pathogens build up in healthy soils? And I kind of this is my deafening silence, mic drop moment. What an oxymoron, right? Can you have healthy soils and soil-borne pathogens? And because just by definition, that soil is no longer healthy because it's not supporting plant growth. But if you looked at all those measurements that I talked about, we look at soil carbon, we look at some of those microbes, we look at microbial activity, you can come back without measuring anything about soil-borne pathogens and be like, yes, this is a very healthy soil. And then I'll be like, well, go put in a diagnostic test for some of these soil-borne pathogens that I can tell you it's not a healthy soil. So sort of this question is, how do we link soil properties with soil-borne pathogen buildup? And so I kind of put that in my little wheel at the bottom there, that if pulses lead to healthy soils, but pulses can sometimes also lead to these soil-borne pathogens. Then we kind of have a disconnect of what's going on between wanting to grow pulses, wanting to grow legumes, but also encouraging the soil-borne pathogens. And so then the big question, and I think why I was asked to speak today, is this question of, can cover crops act as reservoirs for soil-borne pathogens? So I'm going to start by talking about root rot of pulses. Like I said, I love pulses, but they're breaking my heart because they really just have this problem with root rot. And I get up and I talk at these events, and I talk to producers, and it breaks my heart every time when I know the difficulty that they're going through with root rots. And really, it's peas and lentils that are have the primary issues, but we see it occur on there's root rots on faba beans, soybeans, and dry beans as well. So I'm just going to get into what some of those pathogens are, so then we can take a deeper look into those and see what the risk is of crossing over into some of the legume cover crop species. Now, I did debut this slide at Farming Smarter, I think last year, and it was such a hit that I use it at all my talks now. But really, this just sums up what our main root rot pathogens are. Aphanomyces euteiches, everybody that knows me knows that that's my, I don't know, I was like going to say my first love, but I guess it's more like my first enemy. My number one enemy, and then after that we have some of our other pathogens like the Fusarium species and then Pythium, other Fusarium species that don't include Avanasium and Solani, and then also some Rhizoctonia species. And I'm going to kind of tease those apart a little bit more as we go on. Now, the big problem with Aphanomyces and why it causes problems to pulse crop and why there's these risks to cover crops is it produces these oospores that survive a very long time in the soil. So we can see in the pictures the little cells that are in these roots, and you can see they have a really thick double cell wall, and then this nice lipid granule in the middle that just lets them sit in the soil and survive. So if you don't put a pea or lentil in there, you know that's what we are recommending, but then you start putting some of these legume cover crops in to build your soil health. Are you helping the problem, or are you potentially making the problem worse? So I'm going to talk about first what the host range is of Aphanomyces on sort of the legume, cash crops, but I have included the legume forages in here as well. So, peas and lentils, they are very susceptible to Aphanomyces. You get a lot of those oospores produced in the roots. Those get left behind in the soil. So definitely peas and lentils, not a good option. Dry bean, it's variable, and I'll show you why on the next slide, and so is alfalfa. So when we see alfalfa in that list of cover crop species, you start thinking, well, is that the best idea if it can be a host for Aphanomyces? And then chickpeas, faba beans, soybean, sainfoin, and fenugreek are resistant to Aphanomyces, so those might actually be good choices. And I would say sainfoin and fenugreek they didn't show up on any of the list of cover crop species, so they're not high up there, but because we have a breeding program at AFC, we did test them, and maybe they're a good option as legumes for cover crops. And then we look at alfalfa, it's a bit more of a complicated story because we tested a whole bunch of different varieties, and you can see the dark green bar showing some have really, really high disease severity, some don't really, some cultivars have fairly low disease severity, but then if you look at the oospores that are produced in the roots, it doesn't always match that disease. So in the top picture there, that's AAC Dalton, and we could see if you look on the graph, it had very high disease severity. It also produced a lot of oospores. But the one below is AC Blue Jay, I think that's the fifth bar there. You can see it had very little disease, but it actually had a fair amount of oospores. So we can get sort of this symptomless infection where it looks like it doesn't have disease, but if you look at pathogen colonization, it's actually there. And then the issue with alfalfa, peas, and beans is this issue of races and pathotypes. So an isolate from pea, if you look across the top row, you can see it can infect pea, it can infect bean, and it can infect alfalfa. But an isolate from alfalfa, the bottom two rows there, if it's race 1, it can infect pea, it can infect bean, and it can infect alfalfa. But if it's race 2, it can infect pea, but it still can infect alfalfa. So it's a little bit complicated of a race structure, and we don't know in Canada whether we have race 1 or race 2, and we don't test our alfalfa varieties for resistance to race 1 or race 2, whereas they do in the US. I think they kind of recognize the Aphanomyces on alfalfa as a problem, whereas we don't really recognize that here in Canada. And it's the same issue with dry bean. You know, I guess we don't tend to think of it as a pulse that's good for the soil because it needs fertilizer, but if we're thinking of starting to incorporate cover crops into some of those dry bean crops to help with soil erosion, then we want to make sure that if we're putting a legume crop in there, that it's one that's going to be safe for beans. And then it's not just Aphanomyces that's the problem. I think people have heard me talk about this as well. So we kind of have our root rotting gang members, and those are our Pythium species, Rhizoctonia, and then this whole slew of Fusarium species. And that's what I'm going to talk about next, because Fusarium species are ones that can have a broad host range, and that maybe are the ones that are actually more problematic than at least Aphanomyces. We know and we can pinpoint the host range. And Fusarium avineseum is the one that's kind of more problematic when we're looking at pathogen kind of carryover and crossover. And this is because it has a very broad host range. Unlike Aphanomyces and actually many other Fusarium species, it doesn't produce a resting spore. So in order for it to survive, it kind of has to like hitch a ride on all the crops that are in the ground, or else it would just die out every winter. And so because of that, we often find it on pulses, we find it on cereals. It's becoming an increasing concern as being associated with Fusarium head blight, and it does produce some emerging toxins on Fusarium head blight infected grain. And it's become increasingly present over the past 10 to 15 years. Like when I started here in 2011 and I did a literature search for Fusarium avineseum, maybe got 100 hits. If you do a literature search now for Fusarium avineseum, you're up in the tens of thousands. So it's just like increasing worldwide everywhere. And I think it's partly because of this no-till cover crop usage, sort of having a continuous sort of green belt or green bridge in fields, right? And that's what it needs to survive. And so what we did was try to look at, okay, well, what are some legume cover crops? If we grow them in a pea root rot infested field, this was actually just about 10 minutes away from the research center in a producer's field. This was just to look at, well, what species do we find on these legume cover crops? So this is a picture of what our peas looked like in this field. So you can see very, very heavily infected with root rot, Aphanomyces root rot. And then we can see that those cover crops actually looked quite good. I think it's a clover, a lupin, and some sort of vetch kind of in that picture. And we can just see from sort of the biomass, the shoot establishment, much, much better than peas. Then if we look at the roots of the different clovers, they all actually looked pretty good. And then compare that to peas that I put up there in sort of the top left corner, you can see what those peas look like in that field. So these roots looked really healthy, but you'll see there's like a little plus, question mark, and a minus, because when we tested them, even though they looked healthy, the ones with the plus had Aphanomyces on them. The ones with the question mark, it was like right at that sort of cutoff level, so we couldn't tell if it was a true positive or a negative. And then the one with the negative, it actually looks like it's got root rot, but we didn't find Aphanomyces on it. And then the vetches, a completely different story. Here we can see that they have fairly significant root rot, very close to the symptoms that we see for Aphanomyces on pea and lentil, and they did all test positive for Aphanomyces. Still not to that same level that we saw in peas, and like that chickling vetch in the middle there just has beautiful nodules, right? That's what we want from our legume crops. And so this just summarizes what we found there. So we looked at both Aphanomyces, Fusarium avineseum, that's why I kind of introduced it before, and then another Fusarium species called Fusarium redolens. And so what we can see is that those vetches, the three species we looked at, had really high levels of root rot, and they supported high levels of Aphanomyces and high levels of Fusarium avineseum. The clover, like I said, those top three didn't really look diseased, but when we tested them for Aphanomyces, we could find them colonizing the roots, but not to the same level as pea, so they're like doing a little bit of damage and probably enough just to sustain their population. And then again, we can see that Fusarium avineseum can colonize those clover species. And then the next four all looked pretty healthy, and they also did not support Aphanomyces growth. So those would be good options for clover species, but they did support some Fusarium avineseum growth. And then none of them really supported Fusarium redolens growth, and that's, Fusarium redolens isn't actually that aggressive on peas either. It's just something that's everywhere in the soil. And then a similar study done in Germany, and this is looking at Fusarium species also present on legume cover crops. They just took it a little bit farther because we only looked at Avinaceum and Redolins, and they looked at some other Fusarium species that are also implicated in the pea root rot complex. And so what they found, you can see, is Fusarium oxysporum was present on the clovers and the vetches, the same with Fusarium avinaceum, and then a little bit of Fusarium solani. So again, just looking at how these Fusarium species kind of have this broad host range that can colonize both sort of our cash or pulse cash crops, and then also these legume cover crops that we're very interested in. So bit of, I guess, just caution there. And then I'm going to kind of switch over now, sort of talked about everything I can about pulses, and then I'm going to cover off just the cereals and the brassicas very quickly, because I'm not a cereal or a brassica pathologist. So in terms of cereals, what I showed in that chart was the sort of the common cover crops in the cereals were oats, fall rye, and barley. So I'm going to talk a little bit about oats and fall rye. I'm not going to talk about barley because, I mean, that's a cash crop in itself, so we kind of know what the risk would be if you grow a cash crop as a cover crop. And so when we talk about fall rye, it's a very, very common system in the US Midwest to plant fall rye before corn or soybean. So this group in the States did this really interesting study in 2022 where they just did a whole bunch of kind of drone imagery, satellite imagery, to look at what the soybean and corn loss was after a cover crop in the Midwest in the United States. And so what they found was there was a 5% reduction in corn yield after a non-legume cover crop, and a 3.5% reduction in soybean, again, after a non-legume cover crop. Now, in this study, they couldn't say for sure what the cover crop were. They could only put it as non-legume or sort of grassy species, but they just assumed that most of it was fall rye because that's the most common sort of cover crop system. And so they theorized that it was because of the end use of the fall rye, the water use that the cover crop needs, and then also because of soil oxygen depletion. But they completely ignored pathogens. And this is a really, really well-known, well-characterized sort of phenomenon that they've been studying in the States. So this paper came out in 2016, and there have been extensive studies on this pathogen carryover from fall rye to corn. It doesn't seem to happen from fall rye to soybean, but from fall rye to corn, it can be particularly problematic. And I think that's because they're both like gramineous species. And so I tried to just take one sort of graph from this extensive body of literature on this system, kind of show what happens. And really what they found is that the Fusarium oxysporum, kind of the graph there on the top left, and then Pythium torulosum, the graph on the bottom left, that those can carry over from fall rye to corn. And so those were much higher on corn planted into a fall rye cover crop than from corn planted into a fallow cover crop. And since this paper was first published in 2016, like I said, there's been extensive studies trying to look at the very, very specific details of how that occurs. So that's one of the risks with fall rye. And then I'm just going to briefly talk about oats. Oats is like, yay, it's the best cover crop when I started looking into this. Most of the pathogens, there's a lot of shared diseases between oats and wheat and barley, but they're different pathogens. They're very specific pathogens to oats, different species, different forma specialis, as we call them. So most of the pathogens are specific to oats and do not cause diseases in other cereal crops. But there hasn't really been any field studies, particularly on sort of pathogen crossover. So this was just me kind of combing through diseases of field crops in Canada. New edition coming out soon, I should add. And then just sort of combing over, okay, what are the different diseases on these crops and trying to find out, okay, which is the one shared one. And the one possibility there is Fusarium head blight. And then I did get in touch with my cereal pathologist colleague and sort of asked him if this could be a concern. He said that it's not a really big concern on oats. You don't see it that much. And so you don't get a lot of inoculum buildup. So I think you would be fairly safe, but I would say use oats after a cereal crop, not before a cereal crop, just to be safe. And often oats are used kind of if you plant them in the fall and they get winter killed anyway, you're not getting to that stage where they would have Fusarium head blight. Okay, then I'm going to switch over quickly and talk about brassicas again. Pulse pathologist, so this is me kind of combing through literature. But we did have a study in Lethbridge, that same one where I showed you with the legumes. We had another study where we did look at sort of the effect of brassica cover crops on pea root rot. And the good news there is that there was no shared pathogens between pulses and the brassica cover crops. What we did find is that brassica cover crops got a lot of flea beetles. So of course you have to, that's a whole other talk, I guess, sort of crossover between insects and your cash crops. But so you maybe have to manage your cover crops for some of the same problems that, say, canola would have. But those brassica cover crops established really well. And we were interested in them because things like radish, turnips, mustards are sort of known to have biofumigant properties. So we wanted to know, well, are they actually going to affect the soil-borne pathogens? We were not able to prove that with this study. Either way, we didn't really see that they had an effect on pea root rot. They just didn't support the pathogens, at least. But so some of the pathogens of concern that could then carry over between brassica and, of course, our number one cash crop, canola, is the big issue in canola, which is clubroot. And so just a quick sort of literature search tells me that clubroot does supposedly infect all species of plants in the brassica family, but I'm going to show you a slide on a graph on the next slide that maybe refutes that a little bit. And then canola also can have root rots. And surprise, surprise, the root rots are caused by Fusarium, Pythium, Rhizoctonia, so the same ones that we find on peas, just not Aphanomyces. So those are sort of our generalist pathogens that are everywhere. And so I found another study from Germany. There's sort of a theme. I think they really like cover crops in Germany because that's where I found a lot of different studies, where they did test a lot of these different brassica cover crops against the clubroot pathogen just to see, again, does it cause disease? And if it can cause disease, is it actually colonized by Plasmodiaphora? And so what they found there is that common wintercress and garden radish, I think you could just see those have two really, really low bars. Those actually didn't get clubroot, and they also tested positive for the clubroot spores, but the colonization was very, very low. So they theorized they might even kind of act as like a trap crop where they're getting those clubroot spores to germinate. They infect, but they're not completing their life cycle. But then a lot of the other brassicas, like white mustard, I don't know what charlock is, camelina, like those all do support clubroot. Okay, and then the last thing I'm going to talk about that doesn't really fall under a crop is my second favorite pathogen, or another pathogen I do a lot of work on, and that's Sclerotinia sclerotiorum. And I think everyone is familiar with this, with both canola and dry bean. And this one doesn't fall under a crop because it can infect all the crops. Yay! It can really infect all the broadleaf crops. So dry bean, canola, potato, mustards, other pulses. You can find it on peas. You can find it on faba bean, flax, hemp, etc. So it really is one of our pathogens that has a really, really broad host range. And so we try to take care of it by rotating with cereals because it doesn't infect cereals. But the sclerotia, I showed the picture down there. There's these little hard sort of rock-shaped things. They can survive for a fairly long time. So if we're kind of having broadleafs are one of our major cash crop rotations, and then we're also putting in cover crops, broadleaf cover crops into the rotation, then what's that going to do to the Sclerotinia population? And I don't know. Again, the same group in Germany that tested the host range of clubroot on cover crops also tested the host range of Sclerotinia on cover crops. They found, I guess not what's unexpected, is that on a lot of these broadleaf cover crops like Phacelia, Camelina, lupin, field pea, vetch, flax, that of course Sclerotinia can infect them because we already know that they're sort of pathogens of those cash crops anyway, right? What was interesting is that they found that for alfalfa and carnation clover, it didn't produce sclerotia, but like seed alfalfa has a problem with Sclerotinia and it does produce sclerotia, so that's, I guess, just what they found in the greenhouse, whereas out in the field, I think you could find sclerotia in alfalfa. But I think what's interesting is that there's a lot of kind of these lab greenhouse studies looking at host range of pathogens, but there's not a lot of field-based studies actually saying, okay, if we have this mixture of cover crop species out there, what pathogens are we finding? Except for that corn fall rye study, which has been extensively studied in the field. There hasn't been a lot of other studies in that area. So I just wrap up by, I guess, coming back to the title of my talk, proceed with caution. I guess as the plant pathologist, I'm always like the doomsday person. But yeah, just be aware of the risks of what some of these plants, what they can host. And particularly with legumes, I would stay away from peas, alfalfa, and vetches in your cover crop mixture because of that risk to Aphanomyces carryover. And even if you're not a pea grower or a lentil grower, if you start using these as cover crops and then you decide down the road you might want to grow peas, you've probably kind of introduced it and built up that inoculum in your soil. Clover, fava bean, and lupins, I'd say would be the safer choice as sort of cover crop mixes. For brassicas, our main concern is the clubroot pathogen. We don't have to worry about it down here, so now I guess you're okay, but I showed that map and it is clubroot slowly spreading, so who knows when it'll be here. Doomsday. Radish did appear to be kind of a safe option there. And I was surprised actually in this sort of graph of all the cover crop species that there wasn't a lot of brassicas included there. I think radish is one of the few brassicas actually included in that list that prairie producers are using. In terms of cereal, I think oats is a fantastic choice. We've actually tested it as well as sort of a preceding crop to peas, and it does provide a benefit. So there's something going on with oats. I think the only caution would be Fusarium head blight, but if you're not growing oats to seed anyway as a cover crop, then you wouldn't have to worry about that problem. And then fall rye, it can harbor some of those, the Pythium species and some of those Fusarium species as well. And then just looking at it from the other side, the pathogens to watch for, I think in terms of those generalist pathogens, Fusarium and Pythium species, they're kind of everywhere, and I guess you have to just be concerned based on what your cash crop is and how damaging they can be to that cash crop. And then Sclerotinia, of course, is sort of another generalist pathogen that can be everywhere. It's particularly for legumes, it's Aphanomyces euteiches that is sort of the main concern, and you don't want to plant a cover crop that can harbor Aphanomyces. And then for brassicas, clubroot would be the main concern, and flea beetles. But like I said, for the most part, this was an interesting topic for Farming Smarter to want me to talk about because it's not out in the atmosphere yet. There's not a lot of research out there beyond these sort of greenhouse lab-based host range tests. People aren't going out into the field into cover crops and being like, okay, well, what pathogens are occurring on these? So I think Yvonne said she wanted to do her next survey in 2025, and I was like, ooh, well, maybe I can go out and look for pathogens when they do that survey. So I think that's kind of the next step that needs to come together. And then with that, when I'm not talking to Aphanomyces, I'm in the mountains. So happy to take questions.
Any questions out there? Time for one or two quick ones. Actually, we're there first. Next, come back here. Roger, you're number 2.
Did you differentiate between cover crops that were full season versus shoulder season ones?
No, and like a lot of these, that's what I said, like a lot of this is just greenhouse sort of lab-based tests, just looking at the theoretical host range, there's not a lot of information from what's actually occurring in the field, right? And so like like oats is saying, well, if you're just using it as a shoulder season, there's no risk of Fusarium head blight because you're not growing it to seed. So for sure, how it's being used will have an impact, yeah.
Yeah, because I've heard that like Ron Howard had given kind of an idea, or he'd thought that there could be there could be a positive where if they're shoulder season and they die before it reproduces, it could help lower disease, but he said there wasn't that research out there. So
yeah,
you didn't come across stuff like that
No.
So that's a big area.
I think this whole like field study of what's happening to pathogens on cover crops is really something that's lacking. I mean, I'll say with fall rye, what they've found is that it's as those roots decay, so when you do the kill like in the spring, as you're planting in your corn or soybean, it's as those roots decay that you're allowing Pythium to grow on those decaying roots, and then it kind of jumps over to the living roots, right? So there's this question of, okay, introducing sort of dying roots at the same time as living roots, but if it was all in the fall and the roots were dead, there was no kind of bridge there, yeah.
A little while back, I had a discussion with some French producers, and they had a pea processing slash protein extraction plant there. Now that it was very viable for them to grow peas, so they pushed the rotation pretty tight. They told me that due to that process, they ultimately ended up with 5-10 bushel yields instead of the 50, 60 or whatever. And they also told me that some of the plant soil diseases stayed in the soil for 3-4 years. Then they mentioned one that stayed up to 30 years. Do you know anything about that?
Yep, that's our friend Aphanomyces. Yeah, it's the same. So that's the same soil-borne pathogen that really sort of, if you look at the yield, the pea yield graphs from France, it was really, really high in the 90s, and then it just like collapsed, and that's because of Aphanomyces. And it's that same soil-borne pathogen that can survive in the soil for a really long time. So yeah, we're having producers now in this area starting to struggle with that.
What is the proper rotation? How many years apart?
Oh. Yeah, so we are actively studying that. We recommend 6 to 8 years, and we've seen some fields, like in the black soil zone, that have recovered after 6 years. We've been in a field down here where we've been doing research since 2016, and the root rot still hasn't recovered. So how many years is that? 8 years? 8 years of field seasons, and it hasn't recovered. So it's, yeah, we don't quite know exactly what that time is. No. So don't put vetches in there, because you're just going to extend it. Yeah.
Hey, thank you very much.
Yes, thank you.
There's a small
Thank you.
appreciation. If you want to join me in thanking Dr. Chatterton.